First week of development. Ovarian cycle. At puberty, the female begins to undergo regular monthly cycles. These sexual cycles are controlled by the hypothalamus. Gonadotropin-releasing hormone, GnRH, produced by the hypothalamus, acts on cells of the anterior lobe, adenohypophysis, of the pituitary gland, which in turn secrete gonadotropins. These hormones, follicle-stimulating hormone, FSH, and luteinizing hormone, LH, stimulate and control cyclic changes in the ovary. At the beginning of each ovarian cycle, 15 to 20 primary stage, preantral, follicles are stimulated to grow under the influence of FSH. The hormone is not necessary to promote development of primordial follicles to the primary follicle stage, but without it, these primary follicles die and become atretic. Thus, FSH rescues 15 to 20 of these cells from a pool of continuously forming primary follicles, Figs 1 and point 2. Under normal conditions, only one of these follicles reaches full maturity, and only one oocyte is discharged, the others degenerate and become atretic. In the next cycle, another group of primary follicles is recruited, and again, only one follicle reaches maturity. Consequently, most follicles degenerate without ever reaching full maturity. When a follicle becomes atretic, the oocyte and surrounding follicular cells degenerate and are replaced by connective tissue, forming a corpus atreticum. FSH also stimulates maturation of follicular, granulosa, cells surrounding the oocyte. In turn, proliferation of these cells is mediated by growth differentiation factor 9, a member of the transforming growth factor 3, TGF-3, family. In cooperation, theca interna and granulosa cells produce estrogens, theca interna cells produce androstenedione and testosterone, and granular cells convert these hormones to estrone and 17-p-estradiol. As a result of this estrogen production, the uterine endometrium enters the follicular or proliferative phase. 2. Thinning of the cervical mucus occurs to allow passage of sperm. 3. The anterior lobe of the pituitary gland is stimulated to secrete LH. At mid-cycle, there is an LH surge that 4. Flavates concentrations of maturation promoting factor, causing oocytes to complete meiosis I and initiate meiosis II. 5. Stimulates production of progesterone by follicular stromal cells, luteinization, causes follicular rupture and ovulation in the days immediately preceding ovulation, under the influence of FSH and LH, the vesicular follicle grows rapidly to a diameter of 25 mm to become a mature vesicular, Graafian, follicle. 6. Coincident with final development of the vesicular follicle, there is an abrupt increase in LH that causes the primary oocyte to complete meiosis I and the follicle to enter the preovulatory mature vesicular stage. Meiosis II is also initiated, but the oocyte is arrested in metaphase approximately 3 hours before ovulation. In the meantime, the surface of the ovary begins to bulge locally, and at the apex, an avascular spot, the stigma, appears. The high concentration of LH increases collagenase activity, resulting in digestion of collagen fibers surrounding the follicle. Prostaglandin levels also increase in response to the LH surge and cause local muscular contractions in the ovarian wall. Those contractions extrude the oocyte, which together with its surrounding granulosa cells from the region of the cumulus oophorus breaks free, ovulation, and floats out of the ovary, Fig. Point 3. Some of the cumulus oophorus cells then rearrange themselves around the zona pellucida to form the corona radiata, Figs. Point 25 to point 6. Corpus luteum. After ovulation, granulosa cells remaining in the wall of the ruptured follicle, together with cells from the theca interna, are vascularized by surrounding vessels. Under the influence of LH, these cells develop a yellowish pigment and change into lutein cells, which form the corpus luteum and secrete estrogens and progesterone, Fig. 3c. Progesterone, together with some estrogen, causes the uterine mucosa to enter the progestational or secretory stage in preparation for implantation of the embryo. Oocyte transport. Shortly before ovulation, fimbriae of the uterine tube sweep over the surface of the ovary, and the tube itself begins to contract rhythmically. It is thought that the oocyte, surrounded by some granulosa cells, Figs 3b and 4, is carried into the tube by these sweeping movements of the fimbriae and by motion of cilia on the epithelial lining. Once in the tube, cumula cells withdraw their cytoplasmic processes from the zona pellucida and lose contact with the oocyte. Once the oocyte is in the uterine tube, it is propelled by peristaltic muscular contractions of the tube and by cilia in the tubal mucosa, with the rate of transport regulated by the endocrine status during and after ovulation. In humans, the fertilized oocyte reaches the uterine lumen in approximately 3 to 4 days. Corpus albicans. If fertilization does not occur, the corpus luteum reaches maximum development approximately 9 days after ovulation. It can easily be recognized as a yellowish projection on the surface of the ovary. Subsequently, the corpus luteum shrinks because of degeneration of lutein cells, luteolysis, and forms a mass of fibrotic scar tissue, the corpus albicans. Simultaneously, progesterone production decreases, precipitating menstrual bleeding. If the oocyte is fertilized, degeneration of the corpus luteum is prevented by human chorionic gonadotropin, a hormone secreted by the syncytio trophoblast of the developing embryo. The corpus luteum continues to grow and forms the corpus luteum OF pregnancy, corpus luteum graviditatis. By the end of the 3rd month, this structure may be 1/3 to 1/2 OF the total size OF the ovary. Yellowish luteal cells continue to secrete progesterone until the end of the 4th month. Thereafter, they regress slowly as secretion of progesterone by the trophoblastic component of the placenta becomes adequate for maintenance of pregnancy. Removal of the corpus luteum of pregnancy before the 4th month usually leads to abortion. Fertilization, the process by which male and female gametes fuse, occurs in the ampullary region of the uterine tube. This is the widest part of the tube and is siose to the ovary, Fig. 4. Spermatozoa may remain viable in the female reproductive tract for several days. Only 1% of sperm deposited in the vagina enter the cervix, where they may sun for many hours. Movement of sperm from the cervix to the uterine tube occurs by muscular contractions of the uterus and uterine tube and very little by their own propulsion. The trip from cervix to oviduct can occur as rapidly as 30 minutes or as slow as 6 days. After reaching the isthmus, sperm become less motile and cease their migration. At ovulation, sperm again become motile, perhaps because of chemotractants produced by cumula cells surrounding the egg, and swim to the ampulla, where fertilization usually occurs. Spermatozoa are not able to fertilize the oocyte immediately upon arrival in the female genital tract but must undergo, 1. Capacitation and, 2. the acrosome reaction to acquire this capability. Capacitation is a period of conditioning in the female reproductive tract that in the human lasts approximately 7 hours. Thus, speeding to the ampulla is not an advantage because capacitation has not yet occurred and such sperm are not capable of fertilizing the egg. Much of this conditioning during capacitation occurs in the uterine tube and involves epithelial interactions between the sperm and the mucosal surface of the tube. During this time, a glycoprotein coat and seminal plasma proteins are removed from the plasma membrane that overlays the acrosomal region of the spermatozoa. Only capacitated sperm can pass through the corona ceus and undergo the acrosome reaction. The acrosome reaction, which occurs after binding to the zona pellucida, is induced by zona proteins. This reaction culminates in the release of enzymes needed to penetrate the zona pellucida, including acrosin and trypsin-like substances, Fig. 5. The phases of fertilization include the following: Phase 1, penetration of the corona radiate. Phase 2, penetration of the zona pellucida. Phase 3, fusion of the oocyte and sperm cell membrane. Phase 1, penetration of the corona radiata of the 200 to 300 million spermatozoa normally deposited in the female genital tract. Only 300 to 500 reach the site of fertilization. Only one of these fertilizes the egg. It is thought that the others aid the fertilizing sperm in penetrating the barriers protecting the female gamete. Capacitated sperm pass freely through corona cells, Fig. 5. Phase 2, penetration of the zona pellucida. The zona is a glycoprotein shell surrounding the egg that facilitates and maintains sperm binding and induces the acrosome reaction. Both binding and the acrosome reaction are mediated by the ligand ZP3, a zona protein. Release of acrosomal enzymes, acrosin, allows sperm to penetrate the zona, thereby coming in contact with the plasma membrane of the oocyte, Fig. 5. Permeability of the zona pellucida changes when the head of the sperm comes in contact with the oocyte surface. This contact results in release of lysosomal enzymes from cortical granules lining the plasma membrane of the oocyte. In turn, these enzymes alter properties of the zona pellucida, zona reaction, to prevent sperm penetration and inactivate species-specific receptor sites for spermatozoa on the zona surface. Other spermatozoa have been found embedded in the zona pellucida, but only one seems to be able to penetrate the oocyte, Fig. 6. Phase 3, fusion of the oocyte and sperm cell membranes. The initial adhesion of sperm to the oocyte is mediated in part by the interaction of integrins on the oocyte and their ligands, disintegrins, on sperm. After adhesion, the plasma membranes of the sperm and egg fuse. Because the plasma membrane covering the acrosomal head cap disappears during the acrosome reaction, actual fusion is accomplished between the oocyte membrane and the membrane that covers the posterior region of the sperm head. In the human, both the head and the tail of the spermatozoon enter the cytoplasm of the oocyte, but the plasma membrane is left behind on the oocyte surface. As soon as the spermatozoon has entered the oocyte, the egg responds in three ways: 1. Cortical and zona reactions. As a result of the release of cortical oocyte granules, which contain lysosomal enzymes. 2. The oocyte membrane becomes impenetrable to other spermatozoa. 3. The zona pellucida alters its structure and composition to prevent sperm binding and penetration. These reactions prevent polyspermy, penetration of more than one spermatozoon into the oocyte. 2. Resumption of the second meiotic division. The oocyte finishes its second meiotic division immediately after entry of the spermatozoon. One of the daughter cells, which receives hardly any cytoplasm, is known as the second polar body. The other daughter cell is the definitive oocyte. Its chromosomes, 22 plus X, arrange themselves in a vesicular nucleus known as the female pronucleus, Fig. 6 and 7. 3. Metabolic activation of the egg. The activating factor is probably carried by the spermatozoon. Activation encompasses the initial cellular and molecular events associated with early embryogenesis. The spermatozoon, meanwhile, moves forward until it lies close to the female pronucleus. Its nucleus becomes swollen and forms the male pronucleus, Fig. 6, the tail detaches and degenerates. Morphologically, the male and female pronuclei are indistinguishable, and eventually, they come into close contact and lose their nuclear envelopes, Fig. 7a. During growth of male and female pronuclei, both haploid, each pronucleus must replicate its DNA. If it does not, each cell of the two-cell zygote has only half of the normal amount of DNA. Immediately after DNA synthesis, chromosomes organize on the spindle in preparation for a normal mitotic division. The 23 maternal and 23 paternal, double, chromosomes split longitudinally at the centromere, and sister chromatids move to opposite poles, providing each cell of the zygote with the normal diploid number of chromosomes and DNA, Fig. 6de. As sister chromatids move to opposite poles, a deep furrow appears on the surface of the cell, gradually dividing the cytoplasm into two parts, Figs. 6f and 75. Clinical correlates. Contraceptive methods. Barrier methods of contraception include 1. The male condom, made of latex and often containing chemical spermicides, which fits over the penis, and the female condom, made of polyurethane, which lines the vagina. 2 other barriers placed in the vagina include the diaphragm, the cervical cap, and the contraceptive sponge. Hormonal methods are another commonly used form of contraception. These approaches provide the female hormones estrogen and/or progestin. These hormones produce their effects by inhibiting ovulation, by preventing the release of FSH and LH from the pituitary gland, changing the lining of the uterus, and thickening cervical mucus, making it difficult for sperm to enter the uterus. Hormonal contraception can be provided through birth control pills, a skin patch, vaginal ring, injection, or implant. There are two types of contraceptive pills. The first is a combination of estrogen and the progesterone analog progestin, the second is composed of progestin alone. Both pills are effective, but one may suit some women better than others for various health-related issues. A male pill has been developed and tested in clinical trials. It contains a synthetic androgen that prevents both LH and FSH secretion and either stops sperm production 70% to 90% OFMN or reduces it to a level of infertility. The intrauterine device LUD is a small T-shaped unit and there are two types, hormonal and copper. The hormonal device releases progestin that causes thickening of cervical mucus to prevent sperm from entering the uterus. AISO, it may make sperm less active and both eggs and sperm less viable. The copper type releases copper into the uterus that prevents fertilization or inhibits attachment of the fertilized egg to the uterine wall. It also helps prevent sperm from entering the uterine tubes. Emergency contraceptive pills, ECPs, are used as birth control measures that may prevent pregnancy if taken 120 hours after sexual intercourse. These pills may be administered as high doses of progestin alone or in combination with estrogen Plan B. Other types of ECPs mifepristone Ru486 and ulipristal acetate ELA act as anti-hormonal agents. AISO, mifepristone is effective as an abortifacient if taken after the time of implantation. Sterilization is another form of birth control. The method for men vasectomy, which prevents the release of sperm by blocking the ductus deferens, the tube that transports sperm from the testes to the penis. The sterilization method for women is tubal sterilization, in which the uterine tubes are blocked or ligated. These procedures for both men and women can be reversed in some cases. Infertility is a problem for 15% to 30% of couples. Male infertility may be a result of insufficient numbers of sperm and/or poor motility. Normally, the ejaculate has a volume of 2 to 6 ml, with as many as 100 million sperm per milliliter. Men with 20 million sperm per milliliter or 50 million sperm per total ejaculate continued are usually fertile. Infertility in a woman may be due to a number of causes, including occluded uterine tubes, most commonly caused by pelvic inflammatory disease, hostile cervical mucus, immunity to spermatozoa, absence of ovulation, and others. Clomiphene citrate, Clomid, is a drug used to increase FSH concentrations to stimulate ovulation. The drug is given early in the menstrual cycle to cause ovulation in women who do not ovulate or who ovulate irregularly. AISO, it is given to stimulate egg development for use in vitro fertilization procedures. 1% to 2% of all pregnancies in the United States occur using assisted reproductive technology, ARTJ. Offspring from these conceptions show increases in prematurity, 2,500 g, very low birth W8, 5% for those over 40. In addition to these relatively low success rates, the technique is also associated with a higher rate of birth defects. To increase chances of a successful pregnancy, 4 or 5 ova are collected, fertilized, and placed in the uterus. This approach sometimes leads to multiple births. The frequency of multiple births depends on maternal age, with a higher incidence in younger women, and the number of embryos transferred. For a 20 to 29-year-old woman and 3 embryos transferred, the risk is 46%. Multiple births are a disadvantage because they have high rates of morbidity and mortality. Severe male infertility, in which the ejaculate contains very few live sperm, oligospermia or even no live sperm, azoospermia, can be overcome using ICSI. With this technique, a single sperm, which might be obtained from any point in the male reproductive tract, is injected into the cytoplasm of the egg to cause fertilization. This approach offers couples an alternative to using donor sperm for IVF. The technique carries an increased risk for fetuses to have Y chromosome deletions and appears to be associated with a higher incidence of birth defects than standard IVF techniques. The main results of fertilization are as follows: I. Restoration of the diploid number of chromosomes. 2 half from the father and half from the mother. Hence, the zygote contains a new combination of chromosomes different from both parents. I. Determination of the sex of the new individual. An X-carrying sperm produces a female, XX, embryo, and a Y-carrying sperm produces a male, XY, embryo. Therefore, the chromosomal sex of the embryo is determined at fertilization. Initiation of cleavage. Without fertilization, the oocyte usually degenerates 24 hours after ovulation. Cleavage. Once the zygote has reached the 2-cell stage, it undergoes a series of mitotic divisions, increasing the numbers of cells. These cells, which become smaller with each cleavage division, are known as blastomers, Fig. 8. Until the 8-cell stage, they form a loosely arranged clump, Fig. 9a. After the third cleavage, however, blastomers maximize their contact with each other, forming a compact ball of cells held together by tight junctions, Fig. 95. This process, compaction, segregates inner cells, which communicate extensively by gap junctions, from outer cells. Approximately 3 days after fertilization, cells of the compacted embryo divide again to form a 16-cell momia, mulberry. Inner cells of the morula constitute the inner cell mass, and surrounding cells compose the outer cell mass. The inner cell mass gives rise to tissues of the embryo proper, and the outer cell mass forms the trophoblast, which later contributes to the placenta. Blastocyst formation. About the time the morula enters the uterine cavity, fluid begins to penetrate through the zona pellucida into the intercellular spaces of the inner cell mass. Gradually, the intercellular spaces become confluent, and finally, a single cavity, the biastocia, forms, Fig. 10ab. At this time, the embryo is a biastocyst. Cells of the inner cell mass, now called the embryoblast, are at one pole, and those of the outer cell mass, or trophoblast, flatten and form the epithelial wall of the biastocyst, Fig. 10ab. The zona pellucida has disappeared, allowing implantation to begin. In the human, trophoblastic cells over the embryoblast pole begin to penetrate between the epithelial cells of the uterine mucosa on about the 6th day, Fig. 10c. New studies suggest that L-selectin on trophoblast cells and its carbohydrate receptors on the uterine epithelium mediate initial attachment of the blastocyst to the uterus. Selectins are carbohydrate-binding proteins involved in interactions between leukocytes and endothelial cells that allow leukocyte capture from flowing blood. A similar mechanism is now proposed for capture of the blastocyst from the uterine cavity by the uterine epithelium. Following capture by selectins, further attachment and invasion by the trophoblast involve integrins, expressed by the trophoblast and the extracellular matrix molecules laminin and fibronectin. Integrin receptors for laminin promote attachment, whereas those for fibronectin stimulate migration. These molecules also interact along signal transduction pathways to regulate trophoblast differentiation, so that implantation is the result of mutual trophoblastic and endometrial action. Hence, by the end of the first week of development, the human zygote has passed through the morula and blastocyst stages and has begun implantation in the uterine mucosa. Clinical correlates. Embryonic stem cells, ES cells, are derived from the inner cell mass of the embryo. Because these cells are pluripotent and can form virtually any cell or tissue type, they have the potential for curing a variety of diseases, including diabetes, Alzheimer and Parkinson diseases, anemias, spinal cord injuries, and many others. Using animal model research with stem cells has been encouraging. For example, Mouse ES cells in culture have been induced to form insulin-secreting cells, muscle and nerve stem cells, and glial cells. In whole animals, ES cells have been used to alleviate the symptoms of Parkinson's disease and to improve motor ability in rats with spinal cord injuries. ES cells may be obtained from embryos after IVF, a process called reproductive cloning. This approach has the disadvantage that the cells may cause immune rejection because they would not be genetically identical to their hosts. The cells could be modified to circumvent this problem, however. Another issue with this approach is based on ethical considerations, as the cells are derived from viable embryos. As the field of stem cell research progresses, scientific advances will provide more genetically compatible cells, and the approaches will be less controversial. Most recently, techniques have been devised to take nuclei from adult cells, example skin, and introduce them into enucleated oocytes. This approach is called therapeutic cloning or somatic nuclear transfer. Oocytes are stimulated to differentiate into blastocysts, and ES cells are harvested. Because the cells are derived from the host, they are compatible genetically, and because fertilization is not involved, the technique is less controversial. Adult stem cells. Adult tissues contain stem cells that may also prove valuable in treating diseases. These cells are restricted in their ability to form different cell types and, therefore, are multipotent, not pluripotent, although scientists are finding methods to circumvent this disadvantage. Adult stem cells isolated from rat brains have been used to cure Parkinson's disease in rats, suggesting that the approach has promise. Disadvantages of the approach include the slow rates of cell division characteristic of these cells and their scarcity, which makes them difficult to isolate in sufficient numbers for experiments. Abnormal zygotes. The exact number of abnormal zygotes formed is unknown because they are usually lost within 2 to 3 weeks of fertilization, before the woman realizes she is pregnant, and therefore are not detected. Estimates are that as many as 50% of pregnancies end in spontaneous abortion and that half of these losses are a result of chromosomal abnormalities. These abortions are a natural means of screening embryos for defects, reducing the incidence of congenital malformations. Without this phenomenon, approximately 12% instead of 2% to 3% of infants would have birth defects. With the use of a combination of IVF and polymerase chain reaction, molecular screening of embryos for genetic defects is being conducted. Single blastomers from early-stage embryos can be removed, and their DNA can be amplified for analysis. As the Human Genome Project provides or sequencing information, and as specific genes are linked to various syndromes, such procedures will become more commonplace. Uterus. At time of implantation, the wall of the uterus consists of 3 layers, 1 endometrium or mucosa lining the inside wall. 2 myometrium, a thick layer of smooth muscle. 3 perimetrium, the peritoneal covering lining the outside wall. Fig. 11, from puberty, 11 to 13 years, until menopause, 45 to 50 years, the endometrium undergoes changes in a cycle of approximately 28 days under hormonal control by the ovarioles. During this menstrual cycle, the uterine endometrium passes through 3 stages, 1 follicular or proliferative phase, 2 secretory or progestational phase, 3 menstrual phase, figs 12 and 13. The proliferative phase begins at the end of the menstrual phase, is under the influence of estrogen, and parallels growth of the ovarian follides. The secretory phase begins approximately 2 to 3 days after ovulation in response to progesterone produced by the corpus luteum. If fertilization does not occur, shedding of the endometrium, compact and spongy layers, marks the beginning of the menstrual phase. If fertilization does occur, the endometrium assists in implantation and contributes to formation of the placenta. Later in gestation, the placenta assumes the role of hormone production, and the corpus luteum degenerates. At the time of implantation, the mucosa of the uterus is in the secretory phase, Fig. 12, during which time uterine glands and arteries become coiled and the tissue becomes succulent. As a result, Three distinct layers can be recognized in the endometrium: a superficial compact layer, an intermediate spongy layer, and a thin basal layer, Fig 12. Normally, the human blastocyst implants in the endometrium along the anterior or posterior wall of the body of the uterus, where it becomes embedded between the openings of the glands, Fig. 12. If the oocyte is not fertilized, venules and sinusoidal spaces gradually become packed with blood cells, and an extensive diapedesis of blood into the tissue is seen. When the menstrual phase begins, blood escapes from superficial arteries, and small pieces of stroma and glands break away. During the following 3 or 4 days, the compact and spongy layers are expelled from the uterus, and the basal layer is the only part of the endometrium that is retained, Fig. 13. This layer, which is supplied by its own arteries, the basal arteries, functions as the regenerative layer in the rebuilding of glands and arteries in the proliferative phase, Fig. 13. Summary: with each ovarian cycle, a number of primary follicles begin to grow, but usually only one reaches full maturity, and only one oocyte is discharged at ovulation. At ovulation, the oocyte is in metaphase of the second meiotic division and is surrounded by the zona pellucida and some granulasa cells, Fig. 3.4. Sweeping action of tubal fimbriae carries the oocyte into the uterine tube. Before spermatozoa can fertilize the oocyte, they must undergo 1. Capacitization, during which time a glycoprotein coat and seminal plasma proteins are removed from the spermatozoon head. 2. The acrosome reaction, during which acrosin and trypsin-like substances are released to penetrate the zona pellucida. During fertilization, the spermatozoon must penetrate 1. The corona radiata. 2. The zona pellucida. 3. The oocyte cell membrana, Fig. 3.5. As soon as the spermatocyte has entered the oocyte, 1. The oocyte finishes its second meiotic division and forms the female pronucleus. 2. The zona pellucida becomes impenetrable to other spermatozoa. 3. The head of the sperm separates from the tail, swells, and forms the male pronucleus, Fig. 6 and 7. After both pronuclei have replicated their DNA, paternal and maternal chromosomes intermingle, split longitudinally, and go through a mitotic division, giving rise to the two-cell stage. The results of fertilization are the following: 1. Restoration of the diploid number of chromosomes. 2. Determination of chromosomal sex. 3. Initiation of cleavage. Infertility is a problem for 15% to 30% of couples and can be overcome using assisted reproductive technology, ART. In vitro fertilization, IVF, involves fertilizing eggs in a culture medium and placing them in the uterus at the 8-cell stage. In some cases, eggs are fertilized by intracytoplasmic sperm injection, ICSI, whereby a single sperm is injected into an egg's cytoplasm. These in vitro techniques are associated with an increased risk for birth defects, prematurity, low birth weight, and multiple births. Approximately 1% to 2% of all live births in the United States occur through ART. Cleavage is a series of mitotic divisions that results in an increase in cells, blastomers, which become smaller with each division. After 3 divisions, blastomers undergo compaction to become a tidy grouped ball of cells with inner and outer layers. Compacted blastomers divide to form a 16-cell morula. As the morula enters the uterus on the 3rd or 4th day after fertilization, a cavity begins to appear, and the blastocyst forms. The inner cell mass, which is formed at the time of compaction and will develop into the embryo proper, is at one pole of the blastocyst. The outer cell mass, which surrounds the inner cells and the blastocyst cavity, will form the trophoblast. The uterus at the time of implantation is in the secretory phase, and the blastocyst implants in the endometrium along the anterior or posterior wall, Fig. 12. If fertilization does not occur, then the menstrual phase begins, and the spongy and compact endometrial layers are shed. The basal layer remains to regenerate the other layers during the next cycle, Fig. 13.